Epigenetics is defined by how our behaviors can change the way our genes work without changing our DNA itself. Key aspects of epigenetics include gene regulation, environmental influence, role in health and disease, development, and aging. DNA can be described as the book. Epigenetics are the bookmarks, highlights, and sticky notes that affect how our body reads it. Molecular mechanisms that underlie many epigenetic phenotypes include changes in patterns of DNA methylation. Referring back to the book analogy, it is like putting sticky notes on certain pages of a book, saying, "Don't read this page." DNA methylation adds tags onto DNA that act like switches that turn genes on and off. Next, we have chemical modifications of histone proteins. We can think of this as rolling up or unrolling a scroll. When it is tightly rolled, you can't read it. When it's unrolled, the text is visible. DNA is wrapped around proteins called histones. Adding or removing chemical groups on these histones change how tightly the DNA is packed. Tightly packed DNA is hard to read. Loosely packed DNA would be easy to read. The last molecular mechanism is RNA molecules that affect chromatin structure and gene expression. This is like having a guide who points to certain sections of a book saying, "Read this part or skip this part." Some RNA molecules don't make proteins, they act as helpers that guide proteins to certain genes. They can tell the cell which genes to activate or silence by changing how the DNA is packaged. Epigenetic alteration can be influenced by environmental factors, which include everything outside your DNA that could potentially affect your traits. Some examples of factors can include diet, stress, psychological state, toxins, disease exposure, physical activity, among other factors. Combinations of genes and environmental factors play a role in each person's unique traits, health, and identity. Where we live brings together many of those factors. Your environment's climate, allergens, air quality, and water quality have an impact as well. We can also consider whether you live in a rural place, urban, or something in between. Where a person lives also determines their access to food, recreational resources, transportation, social support, work and school environments, and medical care. All of these factors are very important in identifying the type of life that a person lives, which can ultimately affect your epigenetics. Life experiences, especially early in life, have long lasting effects. For example, studies have found that maternal diet during pregnancy has a major influence in the developing fetus and its long term gene expression. Which brings us to the importance of nutrition. Nutrition is one of the most powerful tools for influencing gene expression. They have the ability to switch the good genes of our DNA as well as have negative effects. Nutrients that affect DNA methylation: folate, B vitamins, choline, methionine. They're all found in green leafy vegetables, eggs, liver, and fish. They provide building blocks for methylation, which is a key epigenetic modification that helps regulate gene expression. Bioactive compounds in foods: compounds that regulate activity of enzymes that write or erase epigenetic marks. Some compounds include polyphenols, which are found in colorful fruits and vegetables, spices, coffee, green tea, olive oil. These can modify histones and reduce inflammation, promoting healthy aging. Sulforaphane is found in broccoli, Brussels sprouts, kale. These are responsible for activating antioxidant pathways and suppresses inflammation. When it comes to fats, sugars, and processed foods, high fat or high sugar diets may create epigenetic changes related to metabolism, obesity, and insulin response. Overall, our daily habits impact how our genes are expressed. In conclusion, food doesn't just provide us with energy. Essentially, what we eat can help shape how our genes behave.